Hello, and welcome to the first lecture of the Bioinformatics Algorithm class. When I started teaching bioinformatics 2 decades ago, one of the biggest challenges was to connect with a very diverse audience. I had to talk to computer scientists and biologists, to physicists and biochemists, to mathematicians and statisticians. The challenge was how to keep computer scientists focused while explaining sometimes convoluted biological concepts. And likewise, how to keep biologists awake while explaining sometimes complex algorithmic idea. I hope that in this class we have an equally diverse audience, and that's why I decided to start from a very simple problem. Here's the first problem in this class. Given a string called text1, copy it to a string called text2. And I see that some of you are already thinking about quitting this class because it's so trivial. Please wait. You might think that it's trivial, but billions of cells replicated right now in your body, your own genome, don't think so. In fact, they're trying to accomplish incredibly complex task of replicating genome. Just think about this. You need to unwrite DNA, 2 strands of DNA, and separate them. You need to make sure that there is enough free-floating nucleotides so replication can complete. Because if you start replication and you have to stop in the middle because there is no energy left, you are dead. So in this course, we will learn about this and many other challenges related to replications. In fact, replication in human genome is so complex that biologists still do not fully understand how it's done. That's why we will focus on replication of bacterial genomes that are 1,000 times smaller. The genome is 1,000 times smaller, and that's why replicating bacterial genome is a little bit easier. I'm not saying that it is trivial. And in fact, right now, there are billions of bacterial cells trying to replicate bacterial genomes inside you, because the number of bacterial cells inside you is 10 times larger than the number of your own human cells. Now, since bacteria usually have circular chromosomes, I have to slightly change my problem. Our new problem is, given a circular genome called text1, copy it to a circular text2. It's equally trivial, but it makes some slight changes in the setup that we need to address. A circular genome doesn't have a start and an end. And that's why roughly 5 million nucleotides E. coli genome has a choice on where to start replication. In this class, we will try to figure out where does it start, because all E. coli cells on Earth start replication in the same region called origin of replication. So our first problem, where in a genome does it all begin? And the only information that we have is the genome itself. Okay? So let's go ahead and let's first try to answer the question of what are the hidden messages in the replication origin that help E. coli genome to figure out where to start. And here's the first, or should I say the third already, problem in this course, finding origin of replication. I'll give you a genome. You have to give me the location of origin of replication in this genome. Does the problem make sense? Yes? Yes? You may be right or wrong, depending on who you are. Are you a computer scientist or biologist? Computer scientist. Computer scientist. Then you are wrong. If you were a biologist, then you could think about an experiment. For example, you can start cutting short pieces from DNA. And when you cut a piece and the cell started suddenly lost ability to replicate, it means that probably you cut out the origin of replication. But if you are a computer scientist, you should just refuse solving this problem. It is an ill-defined computational problem. So how can we turn this ill-defined computational problem into something that makes sense? Let's try to ask a question. How does the cell know to begin replication in short ORIC region? There must be some hidden messages in the genome that tell the cell, start replication right here. What are these hidden messages? But let's first try to formulate the problem right. So the right formulation will be the hidden message problem. In this case, input is a string text representing replication origin, and output is a hidden message in the text. Is this problem clear? If you feel this problem clear, please raise your hands. 1, 2, 3, 4, 5, 6, 7, 8. This problem is absolutely unclear. It is equally poorly defined as the first problem I showed you. Indeed, Sean is not happy again because what is the hidden message? Have I described it? No. So for a computer scientist, there is no even starting point to address this problem. But let's go 2 centuries back, and let's see how somebody in a somewhat similar situation tried to solve a similar problem. We go to the Gold Bug short story by Edgar Allan Poe. And in this story, the hero, whose name was Legrand, tried to decode the message left by pirates. And his hope was that when he decodes the message, he will be able to find the pirate treasure. So he was quite motivated. The only thing he saw is this text. How would you tell what is written, what is coded in this text? What Legrand noticed that a combination of 3 symbols, semicolon, 4, and 8, appears surprisingly frequently in this text. Does it suggest an idea on what's written in the message? Here's the hint. The message is in English. And here's another hint. There is the most frequent word in English. And that's why he was able to substitute semicolon 48 for THE, and this message started to make sense. And if you have time, you can complete decoding. Now, if our original hidden message problem was clearly ill-defined, let's now try to redefine it so it would make sense even for computer scientists. How would we redefine it? Well, for various biological signals, certain words appear surprisingly frequently in the text. For example, at this slide, AATTT appears surprisingly frequently in a short text. It's unlikely that it would happen by chance. And then we formulate our first real Rosalind problem. Frequent words problem is I give you a string and an integer k. And you need to find all most frequent k-mers in text, which mean all most frequent strings of length k in the text. Does the problem clear now? Let's ask Sean. He feels better, but he's still not clear because he doesn't know what exactly the most frequent word means. I think he's a little bit picky today because it's easy to define what the most frequent word means. A k-mer, a string of length k, a k-mer pattern is the most frequent word in a text if no other k-mer is more frequent than pattern. And now even Sean is satisfied, and I wanted to thank Sean for great help with his development of this lecture. Now, the Sean is satisfied, but we should ask biologists whether this problem actually makes sense. Remember, bioinformatics is about connecting computational and experimental ideas. And if we check with biologists, it also looks that they are quite satisfied with this problem. Indeed, replication is performed by DNA polymerase. But to initiate replication, DNA polymerase needs a protein called DNA-A. DNA-A binds to a short, typically just 9 nucleotide segments within the replication origin that is known as DNA box. A DNA box is actually a hidden message that we are looking for. And DNA box essentially tells to DNA protein, bind right here. A DNA protein wants to see multiply DNA boxes to bind efficiently. So it looks like we brought computational people and biologists on the same page, and we are now ready to try to solve this problem. And since it's the first problem at Rosalind, we expect a wide variety of different algorithms proposed by you. The simplest algorithm probably is this one. Start from the first chimer in the text. Slide it through the text and see how many identical chimers are present in the text. How much time it takes? It takes time roughly equal to text multiplied by k. But you need to do it for every chimer in the text, and there are roughly text of them. So overall running time of your algorithm, this very naive algorithm, is text square times k. If text is 1 million, you may have problems. But in our case, text is typically small. The average length of replication origin is just 500, maybe 700 nucleotide. In this course, we will focus and we will soon learn much more sophisticated algorithm. For example, some of you may be amazed that the same task can be done incredibly fast. And we will cover this algorithm in the class. And now we turn to the next topic, which will reveal us that some hidden messages are more surprising than others. After we implemented the frequent words problem, let's apply it to real genomes. And let's start from the replication of origin in the bacterium VIBRIO cholerae. And when we run our algorithm, it turns out there are too many potential replication origins. We actually find 4 frequent words. Not to mention that we don't know yet whether it's even statistically surprising to find a 9-mer in a string of length 500 that appears 3 or more times. So which of these 4 represents a real DNA box, and which of them are just statistical artifacts? Well, in this case, it's actually possible to figure out which of these 4 candidates represent real DNA box, because 2 of them are very special. They represent complementary, reverse complementary chimers. And here I showed that one of them actually is a perfect reverse complement to other. You may think that I'm cheating, because if you look at the red 9-mers in the box, shown in the box, and the red 9-mers shown in the bottom side of this slide, they are different. But remember, DNA has direction, and strands run in opposite directions. So when we look at the string starting from TAC, red string, we should actually read it in the opposite direction, and then everything would make sense. So hidden message found. And it is extremely surprising, almost unbelievable, to find one word appearing 6 times, either as itself or as a reverse complement in a short string. So it reinforces our belief that we are on something. And after we've done it, let's try something else. Let's try to find hidden message in THERMOTOGA PETROPHILA, which is a bacteria enjoying life in a very hot environment, about 80 degrees Celsius. Maybe we should simply look for these 2 chimers that we found before, but unfortunately, they are not even there. So it looks like we need to run our frequent words problem again, and in this case, we actually find 6 frequent words, and which tells us that probably different genomes have different hidden messages. It complicates our life quite a lot, because for thousands of bacteria, we need to search for new and new hidden messages. But with this frequent words problem, we are at least armed with these algorithms to do it for any bacteria. Now let's consult to choose which one of these 6 9-mers represents the real DNA box. Let's consult ORIC Finder software, which tells us that it looks like the one shown on this slide. And we learned, therefore, how to find frequent words and hopefully DNA boxes in any bacterium. But wait a second. I told you how to find DNA boxes as frequent words if origin of replication is known. But of course, the position of the origin of replications is 500 nucleotide regions where we look for frequent words is unknown. How possibly we can find it? Remember, we have to find these 500 nucleotide regions in genomes that may be millions of nucleotides long. And let's resort to the idea that I call clumps or clusters of hidden messages to solve this problem. So what we were doing before, we assume that replication origin regions, maybe 500 nucleotide long, is known. And in these replication origins, we were finding frequent words. For example, in this window, we found 4 frequent words that appear 4 times. So it gives us a hint that maybe it is a DNA box. But if we don't know replication origin, what should we do? Maybe we should look at all possible windows in the genome. And if in a particular window we find some frequent words appearing many times, it's a hint that maybe we found a replication origin. Well, let's define short as something of length L, and many is at least T times. And that defines the notion of LT clump. It's a chimer that appear in a window of length L at least T times. And now you have a new Rosalind challenge, which is clump finding problem. You are given a string called genome, an integer k, the length of chimer, an integer L window length, and T, number of patterns in a clump. In output, you have to find all LT clumps in the genome. And we hope that every LT clump will point us to the location of replication origin. Let's assume that you solve it, and let's run it. Let's run it on E. coli genome, the workhorse of bacterial genomics. Here's the result. We find about 2,000 potential 503 clumps, which means 500 length window where there are frequent words appearing 3 or more times. It is just too many. It's absolutely unclear how to choose the real replication origin among them. What should we do? Should we give up? Let's try to see. Let's try to develop new ideas and see how we can move further despite the fact that we seem to be stuck.